Hey biologist, I'm here in the kitchen working on the first part, activity 1, how do polar and nonpolar interactions affect protein structure and properties? And the overarching question for this series of labs is, why is a body temperature of 107 degrees Fahrenheit deadly? And the one we're working on right now is, how do nonpolar and polar interactions affect protein structure and properties? Now, if we think about that overarching question about why a high body temperature is deadly, that high body temperature might affect our proteins. So let's dive into this lab here. In the previous investigation on oil and water, we saw how polar and nonpolar interactions cause a protein to fold into a 3D structure. We talked about that in the activity 3 for the last series of labs. In this activity, we'll further investigate how polar and nonpolar interactions between proteins or between proteins and other small molecules affect the structure and properties of proteins. So you might remember in one of our previous labs that when we have polar and non-polar amino acids together in a chain, I have my yellow kind of beads in this chain here that represent non-polar, and the green represent polar. And as this folds, the yellow parts, the non-polar parts, tend to get smashed into the middle of the protein. So the green parts here can be on the outside and interact with water. So I'm kind of smashing those yellow parts, and it's not that the yellow parts get together, it's that the green parts kind of push them in as they fold, as this protein folds, so that the green parts can be outside interacting with the water, and then the yellow parts, the non-polar parts, get pushed to the inside of the protein. So non-polar yellow is on the inside and green polar is on the outside. So that's something to remember as we go along in this lab. Another thing we want to talk about is what happens when we put pineapple in Jello. We did an experiment in class, how adding fresh and boiled pineapple affects gelatin. I just used Jello brand gelatin. And we need to think about what might be happening at the molecular level to cause the differences we see. I followed the instructions and I'll show you what happened. So we have Jello cam on right now, and this is the control. Sorry, all the writing is backwards. The control is just plain old regular Jello out of the box, made according to the directions. And you can see after it spent some time in the refrigerator, if I put a quarter on top, the quarter stays on top. Even if I wiggle it a little bit, that quarter is going to stay in there. If I wiggle the Jello, you can see it's pretty solid. Doesn't really move when I tip over the container. Now, the next one I did was boiled pineapple. This is boiled pineapple. I boiled it for 10 minutes. And if I put a quarter on top of the boiled pineapple, it's not going anywhere. It's going to stay right where it is. The boiled pineapple Jello, I can tip over like this. Doesn't really move. The fresh pineapple Jello is a different story. If I tilt this container, you can see that this is still pretty much a liquid. And the quarter I tried to put in the bottom of it is literally on the bottom of the jar. I'll put in another one so you can see what happens when you try to put a quarter on this Jello. I'll lower it in with a fork and it just falls right to the bottom. You can see it on the side of the jar there. Up, and it just splashed right in. So I could basically drink this because it's really just a liquid. That fresh pineapple Jello did not solidify in the fridge, and it spent the same amount of time in the refrigerator as the boiled pineapple Jello and the control Jello. So that's the experiment we did, and that's what we're going to have to think about for question 1 in the lab. So we looked at our Jello, and we found that the Jello made with fresh pineapple is acting very differently than the Jello made with boiled pineapple or the control Jello, those Jellos got solid and the quarter stayed on top, where you can see the quarter is on the bottom here, and the fresh pineapple just sunk right in, because this is pretty much a liquid. So you can explain what you observed in the pineapple and Jello demonstration here. And we didn't have a class discussion, but what do you think might be happening to cause that? And I'll give you a hint here. We have our protein that's all folded with the nonpolars in the middle and the polars, and it's very organized. It has a particular shape, but when we heat molecules, we know that they move faster. And some of the connections that normally form when things are a little bit cooler, the molecules might stop connecting and they might not fold quite the same way. So I have a different model here that's kind of stuck together with Velcro here in the middle to illustrate how parts of the protein, how the side chains bond, how some of the polar and non-polar amino acids interact with each other. But as it's heated up, that Velcro tends to come undone. And the protein can change its shape. And when a protein has a different shape, we're going to learn about how its function may change. When it has a different shape or a different form, it may have a different function. So you can put that for question number 2. Gave you a big hint there. So for question number 3, we need to explore this simulation here with pairs of molecules. We have 3 pairs of molecules. One is low polarity. We're going to try to pull them apart, and we can see how difficult they are to pull apart by how far this string stretches. And that one didn't stretch maybe like a third of the screen. Didn't stretch very far. Let's compare that to a very high polarity molecule. You can see the plus and minuses are lining up here because polar molecules, those plus and minuses, they're electrostatically charged and they act like little tiny magnets. And so when we pull here, there's about a third of a screen. That's how hard we had to pull for the 2 low polar molecules. Now that we have high polarity in both our molecules, we have to stretch this spring almost all the way across the screen before they come apart. Let's reset that and do it again. So I'm stretching almost all the way across the screen before they come apart. So it takes more force to pull those high polarity molecules apart. Let's check medium polarity. There's not as many polar atoms here. And we can pull it a little bit farther than we did for low polarity, but not quite as far. We don't need to put quite as much force into pulling the medium polarity molecules. And let's just take a look at these molecules. Notice there's only a couple of polar atoms here. There's more polar atoms here in the medium polarity molecules, and in the high polarity molecules, everything is polar. It's all pluses and minuses, and we really have to pull hard. So question 3, you should know how to answer at this point. How does polarity affect the strength of interactions between the molecules? Remember how far this spring right here, connected to the star, how far that's stretched represents how much force or how stuck together those molecules are. The more you had to stretch the spring, the more stuck together the molecules were. Okay, question 4. Predict how polarity of proteins will affect the types and strengths of interactions the protein can form with other molecules. So justify your answer using ideas from the simulation. So if we have a very polar protein, the green here represents polarity. If we have a lot of polarity in our protein, is it going to potentially interact strongly with other polar molecules? That's what this question is asking. is it going to act like the two molecules up here in the simulation? If you have a lot of polar atoms, remember we talked about how nitrogen and oxygen create a lot of polarity in amino acids and other molecules. If you have those polar atoms in your molecule, how is that going to help make it react with other molecules that also have polar inducing atoms? Atoms that don't share their electrons well. So that's question number 4. If you've got a lot of polarity in your protein, how is it going to behave compared to a protein where you don't have so much polarity? Question number 5, predict how the energy of the system will change when molecules of similar polarity are pulled apart. Justify your answer using the ideas of charge. So if you have a lot of polar molecules, how much energy are you going to have to put into this system to pull them apart versus low polarity molecules that come apart pretty easily? You can play with the simulation more to come up with some ideas for question number 5. Question number 6, predict whether the total change in energy of the system will be different when pulling apart molecules of low, medium, and high polarity. Now remember, we also looked at potential energy of polar and nonpolar molecules in some of the previous labs, and when we separated the polar molecules, the potential energy went up because those molecules had the electrostatic forces that were pulling them together. When they got together, those electrostatic forces weren't there anymore and the potential energy went down. So when you separate those polar molecules, remember there's electrostatic forces pulling them together, and that creates potential energy. When they get together, the potential energy goes down. So we can look at this. If we pull two high polar molecules apart, that we have to put in a lot of energy, and it also creates a lot of potential energies because these two want to get back together. There's a lot of electrostatic forces. There's a lot of pluses that want to line up with those minuses and get back together. So there's a lot of force. There's a lot of potential energy waiting to happen there. So you can answer question 6 on your own using the information I just gave you. Pause the video, answer, and then we'll move on. So, how does polarity affect potential energy in a system of molecules? Explore the simulation and answer the questions below. As you do the simulation, think about how this energy is going to change as polar and nonpolar interactions form within a protein. So if we pull two nonpolar molecules apart, potential energy goes up a little bit, and it goes down when they get back together. Let's do this a couple of times. Watch the potential energy. We're working on question number 7 here. Using the simulation, describe how the energy of the system will change as the two molecules of similar polarity are pulled apart. Explain your answer. So when I pull these apart, it goes up a little bit, then goes back down. Goes up to about 3 bars, that goes back down when they get together. Let's look at the nonpolar ones. When I pull them apart, it's about 2.5 bars. There's a little bit of difference there, so write your answer. You can also work with the simulation yourself. Question 8, predict how changes in the energy of the system affects the stability of the molecules of similar polarity. So if we pull these apart, they're probably going to want to get back together a little bit. There's a little bit of potential energy. This is pretty stable. There's a little bit more potential energy if we pull them apart, but it certainly looks like this protein or this molecular interaction is going to be far more stable if it's together. So this is polar. Let's look nonpolar. pull apart the nonpolar ones, and they'll get back together, but it's a little bit slower. So there's probably, we know that there's a little bit less potential energy here than there is here. And those are going to go boom, right back together. So question 8, predict how the energy of the system will change when polar regions of the protein interact with other polar regions. So if we have a protein here, remember proteins are made of amino acids. We have some polar amino acids represented by green. We have some non-polar amino acids, chunks of the chain here that are represented in yellow. If we have polar amino acids, those polar amino acids are going to want to get out to be with water. The yellow non-polar amino acids here are going to kind of get shoved into the middle. Those green amino acids that are polar want to interact with water and each other, so they're going to come to the outside. Those non-polars are going to get shoved to the inside. The green polars are going to want to interact with each other and with water. So there's a system to how proteins fold depending on the polarity of the amino acids, and there's also some other factors as well, including side chains. So if your protein is nice and stable, it means all the polars are interacting with the polars. If you have an all or a mostly non-polar protein, what's going to happen? Are there going to be equal forces trying to shove everything where it needs to go so the polar parts can interact? It's a good question. Think about it when you answer question number 9. Imagine if a protein had more polar or more non-polar, how that might be different for the protein, and how might those interactions of proteins, the protein with water and the protein with other polar molecules in the human body, or whatever body it's in, maybe it's in a zebra body, how that protein would interact with other polar molecules. If it's mostly non-polar, it might be different. So this gets interesting here. We have lots of polarity and some neutral sections as well here. We have this is the structure of a protein all curled up, and we're going to learn about that. How do polar regions of a protein interact with other molecules? The simulation shows a protein located in the membrane with a small pocket called an active site. So right here, this is called an active site. An active site is a region of a protein with specific shape that can interact with various small molecules, these are our small molecules, in a specific way to carry out a biological function. Your body does a lot of biological functions. All of your cells are doing a lot of biological functions with proteins like this. In the simulation below, there are 3 small molecules next to the protein. 1, 2, 3. Only one of the 3 molecules can form a stable electrical interaction with the protein. The protein and its partners are colored by charge. Red is for negative, blue is for positive, and white-gray is for neutral. So the red is for positive, blue is for negative, and white is for neutral. And remember, negative and positive want to get together, so blue wants to be with red. The protein, your job is to make a prediction about which small molecule, number 1, number 2, or number 3, which one of these, will have the strongest interaction with the protein, and explain your prediction using ideas of charge and polarity. So drag the molecule you think would form the strongest interactions with the protein and place it where you think it interacts best. Include a snapshot of the proposed structure with your answer and explain your choice. So I'll walk you through the first one here, question number 10. Let's pull over number 1. Number 1's got some blue to interact with the red. It fits pretty well, kind of like a puzzle piece. That's kind of what we're looking for. Red is interacting with blue here, so that would seem like it would be pretty sticky and stick together, because we know negative and positive like to get together. Let's put number 2 in there for comparison. it doesn't fit as well, and like the blue and red aren't lining up as well. Let's check number 3. It's got a better puzzle fit shape here, but here we have red with red and we have blue with blue. So it's those negative charges are going to repel each other, and these positive charges are going to repel each other. So that's probably not going to work. I'm thinking that number 1 is probably our best option. So I'm going to take a snapshot here. And my answer is going to include, I'm just giving you an outline here, charges, how the charges align, and also the shape. So make sure your answer includes the word charges, and it also includes the word shape or shapes. And I'll let you do your own screenshot. You can pause the video and do your own screenshot. Question 11 says, which molecules will form the weakest interaction with the protein? Take a snapshot of the proposed structure. Well, you can think about, I'm going to take one out here. That's probably the strongest. Which one's going to be the weakest? Which one looks the least like number 1? And you can also think not only shape, but also think about charges. For example, this one has a lot of neutral. This one has red, where there's red here, so it's got opposing charges. It's got the same charges where they're going to repel. And it's got blue where there would be blue. So these would actually repel each other. So whether it's going to stay in there and be stable is questionable. So you figure out what, based on the shape and based on the charges, which one is going to be the worst fit and make the weakest interaction. And then which one is the medium interaction? And again, base your choice again on shape of the molecules and the charges, because those are both things that affect the stability of two molecules interacting. So pause the video and take snapshots for question 11 and 12, and of course explain your answer in terms of shape and charge. Question number 13. Use ideas from the simulation. Construct your own explanation of how the shape of the molecules affects interactions. Explain your answers using idea of forces. So those electrostatic forces between like charges are going to repel. Opposite charges, positive to negative, are going to stick together. And neutral charges just aren't going to stick very well to either charge. Think about distance. Distance affects the force, the size of the force created by the charge. Think about having two magnets far apart. You can't really feel the electrostatic magnetic force. If you put them together, they go, and you can kind of feel that force get larger as they get closer to each other. And then electrical interactions. That's positive with negative. So this is the first part of this lab, and there will be another video to walk you through other parts of the lab, but you can get the first part of lab 8.5 done.